phenolic compounds. It is an important constituent of woody plants and in association with cellulose to conifers rigidity on them. The decomposition of lignin in nature occurs almost exclusively through the action of certain basidiomycetes called wood-rotting fungi. Two types of wood rots are known: brown rot, in which the cellulose is attached preferably and the lignin left unchanged, and white rot, in which both cellulose and lignin are decomposed. The white rot fungi are of considerable ecological interest because they play such an important role in decomposing wood material in forest. Mushrooms are filamentous basidiomycetes that form large fruiting bodies. The edible part of the mushroom we will discuss in section. The commercial growth of mushrooms as food sources. During most of its existence, the mushroom fungus lives as a simple mycelium, growing in soil, leaf litter, or decaying logs. However, when environmental conditions are favorable, usually following periods of wet and cool weather, the fruiting body develops, beginning the first as a small button-shaped structure underground and then expanding into the full-grown fruiting body that will be above ground. Sexual spores, called basidiospores, are formed bone on the underside of the fruiting body on flat plates called gills, which are attached to a cap of the mushroom. The mushroom basidiospores are dispersed by wind and eventually light on favorable, usually moist, and organic-rich soil and begin the cycle again. Unicellular fungi yeast. The yeast are unicellular fungi, and most of them are classified with an asomycetes. Yeast cells are usually spherical, oval, and cylindrical, and cell division generally takes place by budding. Process of new cell forms as a small outgrowth of the old cell. The bud gradually enlarges along and then separates. Although most yeast reproduce only a single cell, under some conditions, some yeast can form filaments, and in these species, certain characteristics may be expressed only by the filamentous form. For example, the filamentous phase is essentially for pathogenicity, and Candida albicans, and yeast that can cause vaginal, oral, or lung infections, and an acquired immunodeficiency aids patient systematic tissue damage. Yeast cells are much larger than bacterial cells and can be distinguished microscopically from bacteria by their size and by their obvious presence in internal cell structures, such as the nucleus. Some yeast exhibit sexual reproduction by a process called mating, in which two yeast cells fuse. Within the fused cell, called the zygote, ascospores are eventually formed. We discuss the sexual cycle of a typical yeast, Saccharomyces, including the important property of mating types. Yeast flourish in habitats where sugars are present, such as fruits, flowers, and the bark of trees. A number of yeast species live symbiotically with animals, especially insects, and a few species are pathogenic for animals and humans. The most important commercial yeast are the baker's and brewer's yeast, which are members of the genus Saccharomyces. The products of yeast fermentation, the original habitats of these yeast were undoubtedly fruits and fruit juices, but the commercial yeast of today are probably quite different from wild strains because they have been greatly improved through the years by careful selection and genetic manipulation by industrial microbiologists. Indeed, S. cerevisiae has been studied as a model eukaryote for many years and was the first eukaryote to have its genome completely sequenced. Slime molds. Slime molds are microbial eukaryotes that have phenotypic similarity to both fungi and protozoa. Like fungi, slime molds undergo a life cycle and can produce spores. However, like protozoa, slime molds are motile and can move across a solid surface rather than rapidly. From a phylogenetic perspective, slime molds are more ancient than fungi and some protozoa, such as the ciliates, but more derived than flagellated protozoa and their evolutionary predecessors. The slime molds can be divided into two groups: the cellular slime molds, whose vegetative forms are composed of singular amoeba-like cells, and acellular slime molds, whose vegetative forms are masses of protoplasm of definite size and shape called plasmodia. Slime molds live primarily on decaying plant matter, such as leaf litter, logs, and soil. Their food consists mainly of other microorganisms, especially bacteria, which they ingest. Of phagocytosis, slime molds can maintain themselves in a vegetative state for long periods, but eventually form differentiated spores-like structures that germinate and once again generate the active amoeboid. Acellular slime molds. In the vegetative phase, acellular slime molds such as Phylocerum exist in the mass protoplasm of indefinite size. The structure is active motile in amoeboid motion, the plasmodium flowering over the surface of the substratum, engulfing food particles as it moves, amoeboid motion, and the result is cytoplasmic streaming. The cytoplasmic flow forward because of the tip of the plasmodium is less contracted and viscous, and thus cytoplasm takes the path of yeast resistance. In eukaryotic cells, cytoplasmic streaming is facilitated by microfilaments, which exist in the thin layer just beneath the cytoplasm membrane of eukaryotic cells. And in acellular slime molds, cytoplasmic streaming occurs in indefinite strands, each surrounded by the thin cytoplasmic membrane. Streaming itself is the mechanism for distributing cellular metabolites. The acellular slime mold plasmodium is genetically diploid. From this mass of protoplasm, a sporangium and haploid spores can be produced. Under favorable conditions, spores germinate to yield haploid cells. The fusion of two swarm cells then regenerate the diploid plasmodium. Cellular slime molds. Dictostelium. Dictostelium discoideum. A cellular slime mold undergoes a remarkable life cycle in which vegetative cells migrate a cell mass, and eventually produce fruiting bodies in which cells differentiate and form spores. As cell dictostelium become starved, they aggregate and form pseudoplasmodium, a structure in which the cells lose their individuality but do not fuse. This aggregation is triggered by the production of two compounds, cyclic adenosine monophosphate and specific glycoprotein, both of which function as chemotactic agents. We discussed the involvement of CAMP in various regulatory systems and in prokaryotes, those cells that are first to produce these compounds. Become centers for the attraction of other vegetative cells, leading to aggregation masses of cells that come together to form a slimy migrating mass referred to as slug. Fruiting body formation begins when the slug ceases to migrate and become vertically orientated. The fruiting body then becomes differentiated into a stalk, and the head cells into the favored end of the slug become stalk cells, and those in posterior and become spores. Cells that form stalk cells begin to secrete cellulose, which provides the rigidity of the stalk. Cells from the rear of the slug swarm up the stalk to a tip and form the head. Most of these posterior cells differentiate into spores. On maturation of the head of the spores are released and dispersed. Each spore then germinates and becomes a vegetative amoeba. The cycle of fruiting body and spore formation in dictyostelium is an asexual process. However, asexual spores called macrocysts can also be produced. Macrocysts are formed from aggregates of amoebas that become enclosed in the cellular wall. Following this conjugation of two amoebas, a single large amoeba develops and proceeds to phagocytize the remaining amoebas. At this point, a thick cellulose wall forms around the giant amoeba, forming the mature macrocyst, which can remain dormant for long periods. Eventually, the diploid nucleus undergoes meiosis to form haploid nuclei that become integrated into two amoebas that can once again initiate vegetative growth. Algae are a large, diverse group of eukaryotic organisms that contain chlorophyll and carry out oxygenic photosynthesis. Algae should not be confused with cyanobacteria, which are also oxygenic phototrophs, but which are bacteria and thus evolutionarily quite distinct from algae. Although most algae are of microscopic size and hence are clearly microorganisms, a number of forms are macroscopic, some seaweeds growing to over 30 meters in length. Algae are either unicellular and or colonial, the latter occurring as aggregates of cells. When the cells are arranged end to end, the algae is said to be filamentous. Among the filamentous forms, both unbranched filaments and more intricate branched filaments occur. Algae contain chlorophyll and are thus green in color. However, a few kinds of common algae are not green but appear brown or red, because in addition to chlorophyll, other pigments such as xylophiles are present that mask the green color. Algae cells contain one or more chloroplast membrane structures that house the photosynthetic pigments. Chloroplasts can often be recognized microscopically within the algae cells by their distinct green color. We discussed the general structure and properties of the chloroplast in section 14.3 and phylogeny of the chloroplast in section 12.27. Examination shows the algae and constitute a phylogenetically heterogeneous group. Although green algae, chlorophyta, and to lesser extent red algae, rhodophyta, are quite closely related to green plants, other algae groups such as the brown algae and diatoms consist earlier lineages. Even less derived are euglenoids such as the algae euglena. Euglenoids show a phylogenetic relationship to flagellated protozoa, and in fact, cells of euglena can spontaneously lose their chloroplast and exist in completely heterotrophic organisms. Red algae are not worthy in that either chloroplasts contain